name is Jenny Arzolweta, second year college, taking a Bachelor of Secondary Education, and I'm going to discuss about transcription and translation. Transcription and translation. Transcription is the first step in gene expression. It involves copying a gene's DNA sequence to make an RNA molecule. Have you ever had to transcribe something? Maybe someone left a message on your voicemail and you have to write it down on paper. Or maybe you took notes in a class, then rewrote them neatly to help your review. Transcription is the process of copying out the DNA sequence of gene in the similar alphabet of RNA. RNA polymerase. The main enzyme involved in transcription is RNA polymerase, which uses a single-stranded DNA template to synthesize a complementary strand of RNA. Especially, RNA polymerase builds an RNA strand in 5 to 3 direction and adding its new nucleotide to the 3 end of the strand. Step 1. So we have the step 1, the initiation. Initiation is the beginning of transcription. It occurs when the enzyme RNA polymerase binds to a region of a gene called the promoter. This signals the DNA to unwind so the enzyme can read the bases in one of the DNA strands. The enzyme is now ready to make a strand of RNA with a complementary sequence of bases. Step 2, elongation. Elongation is the addition of nucleotides to the mRNA strand. RNA polymerase reads the unwound DNA strand and builds the mRNA molecule using complementary base pairs. There is a brief time during this process when the newly formed RNA is bound to the unwound DNA. So during this process, an adenine A in the DNA binds to an uracil in the RNA. Step 3, we have the termination. Termination is the ending of transcription and occurs when RNA polymerase crosses a step sequence in the gene. The mRNA strand is complete and it detaches from DNA. Stages of transcription. Transcription of a gene takes place in 3 stages. We have the initiation, elongation, and termination. Here, we will briefly see how these steps happen in bacteria. You can learn more about the details of stage and about how eukaryotic transcription is different in the stages of transcription article. Stages 1. The initiation. RNA polymerase binds to a sequence of DNA called the promoter, found near the beginning of a gene. Each gene or group of transcribed genes in bacteria has its own promoter. Once bound, RNA polymerase separates the DNA strands, providing the single-stranded template needed for transcription. The promoter region comes before and slightly overlaps with the transcribed region whose transcription is it specifies. It contains recognition sites for RNA polymerase or its helper proteins to bind to. The DNA opens up in the promoter region so that the RNA polymerase can be transcription. Stages 2. Elongation. One strand of DNA, the template strand, acts as a template for RNA polymerase. As it reads this template one base at a time. The polymerase builds an RNA molecule out of complementary nucleotides, making a chain that grows from 1 to 3. The RNA transcript carries the same information as the non-template coding strand of DNA, but it contains the bases uracil, U instead of thymine. RNA polymerase synthesizes an RNA transcript complementary to the DNA template strand in the 5 to 3 direction. It moves forward along the template strand in 3 to 5 direction, opening the DNA double helix as it goes. Synthesized RNA only remains the bound to the template strand for a short while, then exits the polymerase as a dangly string, allowing the DNA to close back and form the double helix. We have the stages 3 determination. Sequence called terminators signals that RNA transcript is complete. Once they are transcribed, they cause the transcript to be released from the RNA polymerase. An example of termination mechanism involving formation of hairpin in the RNA is shown below. The terminator DNA includes a region of RNA that forms a hairpin structure followed by a string of U-nucleotides. The hairpin structure in the transcript causes the RNA polymerase to stall. The U-nucleotides that come after the hairpin form weak bands with the A nucleotides of the DNA template, allowing the transcript to separate from the template and ending transcription. So we have the three main steps to the process of DNA transcription. We have the initiation. RNA polymerase binds to DNA. DNA is transcribed by an enzyme called RNA polymerase. A specific nucleotide sequence tells RNA polymerase where to begin and where to end. RNA polymerase attaches to the DNA at a specific are called the promoter region. The DNA in the promoter region contains specific sequence that allow RNA polymerase to bind to DNA. So moving forward, we have the elongation. Certain enzymes called transcription factors unwind the DNA strand and allow RNA polymerase to transcribe only a single strand of DNA into a single-stranded RNA polymer called messenger RNA or the mRNA. The strand that serves as the template is called the antisense strand. The strand that is not transcribed is called the sense strand. Like DNA, RNA is composed of nucleotide bases. RNA, however, contains the nucleotides adenine, guanine, cytosine, and uracil. When RNA polymerase transcribes the DNA, guanine pairs with cytosine, and the adenine pairs with the uracil, or the EU. Moving forward, the term of termination. So the last step of the process of transcription, we have the termination. RNA polymerase moves along the DNA until it reaches a terminator sequence. At that point, RNA polymerase releases the mRNA polymer and detaches from the DNA. Translation. Take a moment to look at your hands. The bone, skin, and muscle you see are made up of cells. And each of those cells contains many millions of proteins. As a matter of fact, proteins are molecular building blocks for every organism on Earth. Ever wonder how antibiotics kills bacteria, for instance, when they have a sinus infection? Different antibiotics work in different ways, but some attacks a very basic process in bacterial cells. They knock out the ability to make new proteins. Steps of translation. Your cells are making new proteins every second of the day, and each of those proteins must contain the right set of amino acids linked together in just the right order. That may sound like a challenging task, but luckily your cell, along with those of other animals, plants, and bacteria, are up of the job. To see how cells make proteins, let's divide translation into three stages. Initiation, starting off, elongation, the adding on the protein chain, and termination, the finishing up. Steps in initiation. In initiation, the ribosomes assembles around the mRNA to be read at the first tRNA, carrying the amino acid methionine which matches the start codon, AUG. The setup called the initiation complex is needed in order for translation to get started. In order for translation to start, we need a few key ingredients. So these include the ribosome, which comes in two pieces, large and small, an mRNA, or the messenger RNA with instructions for the protein will build, an initiator, the tRNA carrying the first amino acid in the protein, which is almost always methionine. So steps 2, elongation. Elongation is the stage where the amino acid chains get longer. In elongation, now the mRNA is read one codon at a time, and the amino acid matching each codon is added to a growing protein chain. But how does the chain actually grow? To find out, let's take a look at the first round of elongation, and after the initiation complex has formed, but before any amino acids have been linked to make a change. So lastly, we have the steps 3, the termination. Termination is the stage in which the finished polypeptide chain is released. It begins when a stop codon, codon UAJ, codon UAA, or the codon UGA enters the ribosome, triggering a series of events that separate the chain from its tRNA and allow it to drift out the ribosome. So after termination, the polypeptide may still need to fold into the right 3D shape, undergo processing, such as the removal of amino acids, get shaped to the right place in the cell, or combined with other polypeptides before it can do its job of functional protein. So it is the differences between the transcription and translation.